These methods can provide mechanistic insights into immune cell-mediated tumor cell cytotoxicity and invasive behavior in a 3D setting. The main advantage of this technique is that this 3D spheroid co-culture model does not require the transfer of spheroids for multiple subsequent assays. Demonstrating this procedure will be Apsra Nasir, a PhD student from our department.
To generate the spheroids, using aseptic technique in a cell culture hood, add 500 microliters of molten agarose into an 81 microwell rubber mold, taking care to avoid bubbles. When the agarose is solidified, carefully flex the rubber mold to pop out the agarose casts into individual wells of a 12-well plate. To equilibrate the casts, add 2.5 milliliters of cell culture medium supplemented with 10% fetal bovine serum into each well, and place the plate into a cell culture incubator for 1 hour. At the end of the incubation, tilt the plate to remove the cell culture medium in the surrounding first, then in the seeding chamber, and carefully seed 190 microliters of the prepared tumor cell suspension drop-wise into each cell seeding chamber. After a 15-minute incubation in the cell culture incubator, add 2.5 milliliters of medium to the outside of the cast, and return the plate to the incubator for 48 hours. To set up a T-cell co-culture, resuspend the appropriate number of T-cells in 190 microliters of an appropriate T-cell culture medium per well, and tilt the 12-well plate to allow removal of the cell culture medium.